The overall goal of this protocol is to facilitate the isolation and purification of S100A12 in its active form so that it can be utilized for antimicrobial assays.
This method can help answer key questions in the microbiology field, such as what is the role of S100A12, an antimicrobial peptide which is produced in response to infection by pathogenic bacteria.
The main advantage of this technique is that our optimized protocol provides high yields of stable, pure S100A12 dimer, which has the capacity to bind and sequester nutrient metals.
Generally, individuals new to this method will struggle because S100A12 forms oligomers of varying sizes under certain conditions, and these oligomers have diminished antimicrobial activity in vitro.
We first had the idea for this method when we began purifying and studying the antimicrobial activity of a related S100A family protein called calprotectin.
Visual demonstration of this method is critical to the size and column final purification steps, and the antimicrobial assays are important to the success of this process.
To begin this procedure, transform competent BL21-DE3 cells as outlined in the text protocol. Next, add 1 to 5 microliters of plasmid to 50 microliters of bacteria in a microcentrifuge tube on ice. Incubate for 20 minutes. After this, heat shock the cells at 42 degrees Celsius for 30 seconds. Incubate on ice for 2 minutes. Then, add 500 microliters of SOC media. Incubate at 37 degrees Celsius on